So welcome everyone to our OpenSim webinar. My name is Jen Hicks. I'm the OpenSim R&D Manager and also the Associate Director of our National Center for Simulation in Rehab Research, which supports the OpenSim project. And I'll be serving as the moderator for today's webinar. I'm pleased to welcome today's presenter. Brian Umberger is joining us from the University of Massachusetts Amherst. And he'll be talking about musculoskeletal modeling to study human bipedalism evolution. So next slide. So OpenSim is a freely available software application for visualizing musculoskeletal structures and simulating movements of humans and animals. So the first goal of our webinar series is to showcase the diverse cutting-edge research that's being performed with OpenSim. OpenSim is also a growing and diverse community of users, so the second goal of the webinar series is to provide a platform so that members of the community can communicate with each other and collaborate. Next slide. So before we get started, a few quick reminders about the format of the webinar. We definitely want to have time to ask and answer questions, but we'll do those at the end of the webinar. And we'll do that via the text-based Q&A panel in your WebEx interface. If you need any additional technical help, you can consult the guide on our website. And so with that, I'd like to go ahead and introduce our speaker. So next slide. So excited to have Brian Umberger join us today. Brian's an associate professor of kinesiology and organismic and evolutionary biology at the University of Massachusetts Amherst. Brian's research is focused on enhancing our understanding of the mechanics, energetics, and control of human locomotion by integrating experimental and computer modeling approaches. Brian's also an OpenSim fellow who's been using OpenSim since 2008, so almost the very beginning. He's published models and algorithms for use with OpenSim that he's provided for others to download and use and extend on the SimPK website. Brian's a veteran of many of our OpenSim workshops and other OpenSim events, and he was also the co-recipient of a pilot project award from the NCSRR. So we're really excited to have you join our webinar series, Brian, and with that, I'll let you take it away.
Thanks, Jen. I'd just like to let everyone know I have a bit of a cold today, so hopefully I won't be coughing too much, and I'll just ask you to please bear with me. Before really getting into the talk today, I wanted to acknowledge the fact that the results and models and simulations I'll be sharing with you today are really the result of a team effort. And a major focus of the webinar today is going to be discussing the development and validation of a chimpanzee musculoskeletal model. I want to highlight two people in particular, my colleagues Matthew O'Neill and former postdoc Lengfeng Li, who played major roles in the development and testing of that model. And I also want to more broadly acknowledge my collaborators and current and former students who worked on this research, as well as our funding sources and groups that have provided other resources. So as you look across the animal kingdom, you can find many different examples of bipedal locomotion, getting around on two legs. But it turns out to actually be relatively uncommon. It's much more typical for animals to get around on 4, 6, 8, or more legs. Our closest relatives, the great apes, are perfectly capable of walking on their hind limbs, but most of the time when they're on the ground, they engage in a quadrupedal knuckle walk. Some of the animals that walk and run most like us are particular species of birds like ostriches, emus, rheas. But as I said earlier, bipedalism is actually the exception, not the rule. And among mammals, humans are unique in that we are the only mammal to walk upright habitually on two relatively straight legs. So there's been a lot of interest over time in how this unique form of locomotion would have evolved in our now extinct human ancestors. So I want to introduce a concept that may not be familiar to everyone, and it's related to the group known as hominins. So hominins include us, modern humans, and all of our extinct human ancestors back to the point at which we diverged with chimpanzees. And so you can see in this diagram how hominins are closely related to chimpanzees and then a little bit more distantly related to gorillas and orangutans. And so if we focus in on the hominin and chimpanzee part of this diagram, you can see here we have modern humans and modern chimpanzees on the right side of this figure. And we shared a common ancestor about 7 million years ago, give or take, somewhere on the order of 6 to 8 million years ago. And in that intervening time, there's actually been many hominin species, some more recent that are part of our own genus, like Neanderthals or Homo erectus, and others further back in time. Australopithecus afarensis from about 3.2 million years ago is the famous Lucy fossil. And then Ardipithecus ramidus even further back, about almost 4.5 million years. And these are just some of the creatures that we actually know a bit about from their fossil remains. There's many others as well. And one of the characteristics that they all had in common, despite their vast differences, is that they were all bipedal. And so as we look back through evolutionary time, we can focus on certain aspects of the morphology and how it's changed to gain insight as to how we evolved our unique form of locomotion. So if you're interested in human evolution, why study bipedalism? Well, if we consider the characteristics that supposedly separate us from the rest of the primates, things like our large brains or sophisticated culture, what we find is that habitual bipedalism actually predates any of these other developments by at least 2 million years. And most anthropologists and evolutionary biologists use the transition to habitual bipedalism as the basal event that defines the beginning of the hominin line and ultimately leads to where we are today. So it's very central to what makes us human, as well as being just fascinating to someone who studies the biomechanics of how modern humans walk and how it is different from other animals. So much modern current research focuses on how human locomotion is similar to or different from that in chimpanzees, being an extant species that we can actually study experimentally. As mentioned earlier, chimps are our closest living relatives, and they're considered facultative bipeds, meaning bipedalism is optional for them. And while, as I pointed out in that video, they're fully capable of doing it, it's something they only engage in maybe around 5% of the time. So it's an activity that they use sparingly. In addition to their very different morphology compared with humans, and I'll point that out a little bit better later on, when chimps walk bipedally, they use a flexed limb posture compared with the relatively straight leg posture that modern humans use. And they have a relatively high metabolic cost of walking compared to modern humans. And so there are a number of characteristics that chimpanzees share with what we believe the last common ancestor would have been like in terms of their structure and how they would have moved about. And so it's definitely not that chimpanzees are a perfect model for the last common ancestor, but rather that they share certain characteristics in their morphology and how they move that make interesting contrast with modern humans and might shed some light on the earlier stages of human evolution. Whenever a new fossil hominin is discovered and described in the literature, you invariably get some comparisons to how it looks compared to modern humans, such as on the left here, and how it looks compared to modern chimpanzees, such as on the right. And it turns out that we have a much better understanding of the functional morphology of modern humans and how that affects the way we walk than what is known about chimpanzees. So there's some value in fundamental studies of structure-function relationships in the chimpanzee postcranial skeleton and how that relates to their locomotion, so that these comparisons can be better informed. And for these types of questions, it turns out that musculoskeletal modeling is really well suited for addressing these types of questions because the morphology itself is represented in the model, and you can either make meaningful comparisons between two different individuals or two different species, or you can also change aspects of the morphology and see what the impact that is on movement. Just give you a more concrete example here. This figure shows a side view of the pelvis in a modern human on the left and a modern chimpanzee on the right. Modern humans have a fairly bowl-shaped pelvis. You can see the iliac blades here from the side view, whereas when viewed from the side, the chimpanzee pelvis is almost flat, so the iliac blades are oriented in the frontal or coronal plane. Chimpanzees also have a much more substantial ischial tuberosity here than what you see in modern humans. And so if you look into the fossil record and you take, for example, the reconstruction of the Ardipithecus ramidus pelvis here, you have what appears to be a fairly human-like ilium with a pretty chimpanzee-like ischium. And so properly interpreting the functional consequences of that will be greatly enhanced by having a better understanding of the morphology of modern humans and chimpanzees and how that relates to the way in which we move. So with that, our general approach is to conduct traditional experimental studies in extant species, so far mostly humans and chimpanzees, although there's other good models such as macaques as well. This involves collecting ground reaction force data, 3D motion of the body segments, electromyography, and these are done in concert with modeling and simulation studies in these same species. So we have a tight connection between the modeling, simulation work, and the experimental data. And then this is done in concert with modeling and simulation studies in extinct human ancestors, where we're unable to collect any experimental data, but we can view our simulation results in the context of the human and chimpanzee data and essentially have brackets on what we should consider realistic or what outputs of the simulation maybe we should trust more and which we should be more suspect about. And so before talking about the chimpanzee musculoskeletal modeling effort in more detail, I just want to give you a specific example, a fairly simple and straightforward example of how we are investigating the function of specific muscles and how that is similar or different in humans versus chimpanzees. And because of the dramatic changes in the shape of the pelvis, presumably over evolutionary time, there's been a lot of interest in how that would have affected the function of muscles that attach to the pelvis, particularly the gluteal muscles. We know the gluteals are very important in locomotion for things like balance, support, propulsion, and I'm going to focus here specifically on the gluteus medius. You have the gluteus maximus and minimus. I'm going to focus on the gluteus medius here. And there's been a lot written about how its functional role presumably would have changed with the reshaping of the pelvis from a more primitive pattern to the more bowl-shaped structure with the iliac blades wrapped around to the side as you have in modern humans. And a lot of this is based on inferences from watching animals, modern humans, chimpanzees, move about, and then knowing something about the anatomy and measuring typically EMG to see when the muscle is active. But with a musculoskeletal modeling approach, you can integrate all that data and also go some steps further. So this is a better view, perhaps, of the human and chimpanzee pelvis, human on the right, chimpanzee on the left, viewed in the frontal plane. So you can see it's much taller in the chimpanzee, and these are to scale, even though a chimpanzee is overall much shorter than a human. And then this is, again, the lateral view you saw earlier with the chimpanzee and the human. And we want to know what are the effects of these differences in morphology for pelvic muscle function in walking specifically. And so these red ovals I've put in show the approximate attachment points of the gluteus medius, and you see they're attached at about the same location on the bone, but because the bone is shaped very differently, presumably there might be some differences in how these muscles function that relate to the development of bipedalism. Now, I know many people who signed up for the webinar are either beginning OpenSim users or have not used OpenSim before, so I just wanted to take a moment to mention that all of the analyses that we'll be talking about from now until the end have mostly been done in the OpenSim software. So this is a software environment for musculoskeletal modeling, simulation, and analysis. has the advantages of being open source and freely available to anyone who wants to use it, and now has a fairly long track record of having robust support from the federal government in the form of grants from NIH and DARPA, and the papers cited down here are ones that specifically describe OpenSim and are not just a specific application that used it, but actually provide information about the OpenSim software, as well as all the information you can get on the OpenSim website. So in conducting these studies, we begin by collecting experimental data on chimpanzees and humans involved in bipedal locomotion. The data collection is, if you're familiar with this in humans, is pretty similar in chimpanzees. The major difference is that in order to have the chimpanzee step on the force plates in the right order, which of course they don't know where they are, these are the 4 boxes in the video, it takes many, many, many more trials than it would in humans, where you can just tell them to step on the force plates. But other than that, the basic aspects of data collection and how we integrate the motion capture data here with our models in OpenSim is essentially the same. This allows us then to create dynamic simulations of walking in chimpanzees as well as in humans. And at the end of one of these simulations, after generating it, you would know what the activation of each of the muscles is. You would have an estimate of what force the muscles are producing, and then you would be able to perform any of a number of different analyses on the simulation outputs that might give you some additional insight into how locomotion is being produced and how that's different or similar between these 2 species. Before proceeding with a muscle-based analysis, we would want to confirm that the predicted muscle activations in the models are in reasonable agreement with the timing, at least, of measured EMG data. And so this is just a small sampling of some of the predicted activations in the top row. And each graph has 3 lines in it. That is 1 trial from each of 3 subjects, or 1 simulation from each of 3 subjects. You can see they have a similar pattern with small differences in shape or magnitude. And then this is average EMG data in the bottom row from 3 individuals as well. And so in the analysis I'm going to be showing you in a moment here, that's based on the gluteus medius, so this middle column during the stance phase. And so there's reasonably good agreement between the predicted activations and when we would expect the muscles to be activated based on the EMG data. So to try and understand how a particular muscle is influencing the motion of the body, we perform what's called an induced acceleration analysis. This is something that can be done within the OpenSim software. And what it does is allows you to identify how any force acting on the skeleton causes a joint, a body segment, or the whole body center of mass to accelerate in any of the potential directions in which it can move. And so this was interesting for us in terms of how the activation of gluteus maximus was influencing the motion of the whole body center of mass in walking. And what you have here on the slide right now, over on the left, is a chimpanzee skeleton just after the lead foot has hit the ground, although we did the same analysis with humans as well. When your lead foot hits the ground, if it's the left foot like here, your center of mass will have a lateral component to its velocity. And if nothing acts to counter that, you would then begin to drift off laterally or potentially even fall over. And so there must be something or multiple things happening to resist that. And what we find is that the forces generated by the gluteus medius at this point in the gait cycle produce an acceleration of a center of mass that is in the medial direction. So I hope I said that correctly a moment ago. You have a lateral center of mass velocity, and contraction of this muscle causes a medially directed acceleration of the center of mass. So that will act to counter this velocity, meaning that this muscle group aids in the sort of the side-to-side balance of your center of mass as you walk forward. And what we found was that this functional role of the gluteus medius was essentially the same in chimpanzees and humans. So despite the attachment of this muscle, or the bone to which this muscle attached being shaped very differently, this functional role seems to be preserved despite very different anatomy. When your lead foot hits the ground and you transition onto it, your center of mass also has a downward velocity. And likewise, you need to do something, or perhaps multiple things, to counter that. Otherwise, eventually you would wind up in a heap on the floor. And what we find is in humans, contraction of the gluteus medius in the early to mid stance phase produces an upward vertical acceleration of the center of mass. So this relatively small muscle group we find is producing a lot of vertical support of the center of mass. But when we do this analysis in chimpanzees, we find that the gluteus medius produces no upward acceleration of the center of mass and actually produces a tiny bit of downward acceleration. And that's because the hip is so flexed that there's actually a little bit of a hip flexion moment generated by the anterior fibers of that muscle. So here you have one functional role of the muscle that seems to be preserved across pretty dramatic changes in pelvic shape, but then another functional role that is very different. So the situation can be fairly complex, and in order to tease out these different roles, it's helpful to have the entire morphology represented. Now, of course, chimpanzees don't wind up in a heap on the floor when they take a step, so they must be doing something else to generate that upward acceleration. And what we've found is that they rely a lot on the large hamstrings muscles and the large quadriceps muscles to generate that support. And that may be related to the much higher cost of locomotion they have. If we can generate vertical support with a relatively small muscle, that will tend to lead to better locomotor economy, whereas if chimps need to activate larger muscles, that's going to incur a greater cost. So to do these analyses, we of course need a model of a human and a model of a chimpanzee, and there are existing, and have been for a number of years now, existing models of the human musculoskeletal system. But when we began this project, there were no models of the chimpanzee musculoskeletal system. So as part of this project, we needed to develop one from scratch. And this slide just shows sort of a high-level overview of the workflow. I'm actually going to go through each one of these steps in a little bit of detail. But our chimpanzee pelvis and hind limb musculoskeletal model has been published in the Journal of Experimental Biology in 2013, for anyone who's interested in getting more details than what I'm going to talk about today. And a freely accessible version of the model is available on the SimTK website, the same place where you access the OpenSim software, for anyone who wants to download it and try it. So the rest of my talk is going to focus primarily on the development and validation and use of this musculoskeletal model. So we began by obtaining a complete skeleton of an adult chimpanzee from the American Museum of Natural History in New York City. And the entire skeleton was CT scanned at the Stony Brook University Hospital, where a number of my colleagues are on this work. And because of our interest in bipedalism, we've initially focused on the pelvis and hind limb, but we do have scans of the entire skeleton, so eventually we will be able to develop a four-limb model and then eventually a whole body model for future work. So each of the skeletal elements was first CT scanned, and then after some initial pre-processing in a mirror in Geomagic, and then some downsampling in MATLAB, we created a representation of each of the bony elements that was imported into the software known as SIM. So SIM stands for Software for Interactive Musculoskeletal Modeling. And it's a commercial product, but it has a number of features that make it very well suited for building musculoskeletal models from scratch. And so that's where we did a lot of the initial construction work before exporting the model for use in OpenSim for doing the types of analyses that I mentioned earlier. So the model includes all of the bony elements in the pelvis and right hind limb, and each of these is connected to adjacent bones with a variety of different types of joints. And so the hip is modeled as a spherical or ball and socket joint. We parameterize this by fitting a sphere to the acetabulum and then another sphere, a slightly smaller sphere, to the head of the femur, and then made the centers of those two spheres coincident. The knee is a 1 degree of freedom revolute joint, but it's not just a pure pin joint. It actually has the translational aspects of the axis of rotation that allow the tibial plateaus to track along the femoral condyles, which have quite a different shape than in a human knee. And then there's also some constraints that cause the patella to track along the groove between the condyles in concert with that rotational motion at the knee. And then the ankle joint is also a 1 degree of freedom revolute joint, but it also is not just a pure mediolateral axis. It has a properly anatomically skewed axis. And when you work mostly with humans, you're used to there being a wealth of data on muscle architecture and joint axis orientations and whatnot. And a lot of that data is lacking when you work with other species. But it turns out that with chimpanzees, while there's not nearly as much data available as there is for humans, there often have been 1 or 2 studies done that have quantified some of these key components you need to create a good model, like the proper anatomical orientation of the ankle joint or muscle architecture data, as I'm going to talk about in a moment. Now, on top of the skeletal framework, we have the musculature of the hind limb. And so in this figure, each of the red lines represents the pathway of a particular muscle or part of a muscle. You can see the hamstrings muscles back here, for example. And there are 35 muscle tendon units represented by 45 muscle tendon pathways. And the discrepancy in the number there is that some muscles with broad areas of attachment, like the gluteus medius, for example, are represented by more than 1 pathway. So you might have an anterior, central, and posterior head to the muscle. And so this accounts for all of the major muscles in the hind limb except for the intrinsic muscles of the foot. And those could, of course, always be added later. In terms of the joints, as we were talking about a moment ago, there is motion of the toes in the model within the foot, as well as separate motion of the first digit, because chimpanzees have an abducted big toe, different than humans. But it certainly would be something someone could do at a later point, is add more detail into the motions of the foot as well as the representations of the intrinsic foot muscles. The muscle attachments were handled in a number of different ways, depending on the complexity of the path of the muscle. So each muscle had an origin and an insertion, as you can see in the figure over here on the left. And for some muscles, a single line segment between origin and insertion was adequate to capture its pathway. For other muscles, we used so-called via points to produce wrapping, such as around the lateral malleolus here. And in other cases, we used wrapping shapes, primarily cylinders and ellipsoids, when there was more complex wrapping, such as here at the knee and particularly at the hip, as I'll show in a slide or two. This is again a case where the muscle attachment sites, the origins and insertions, there's quite a bit of data to go on, given that we're talking about a non-human species here. So there are published muscle maps that identify the average areas of attachment for all of the muscles in the chimpanzee musculoskeletal system. Because of our high-resolution scans, we could also see the markings on a lot of the bones that identify where muscles attach. And then when there were questions we had about maybe where the path of one muscle lies relative to another, we were able to conduct some cadaveric dissections to help resolve uncertainty with that. And that was useful in terms of the designing of wrapping functions and the placement of via points, which change the pathway of the muscle at the appropriate point to have it, say, wrap around a bony prominence, or to make sure that 2 muscles don't cross through each other, which of course is not anatomically realistic. So the joint ranges of motion were set up to allow as much range of motion as was possible in the model while allowing realistic muscle pathways. Chimpanzees have a considerably larger range of motion at their hind limb joints than humans do in the lower limb, and so it was important to try to maximize this as much as possible. But if you allow too much range of motion, then it becomes very difficult to model the muscle pathways properly. And so there's a bit of a compromise made there. The ranges of motion from minimum to maximum value that we have here were based on manipulations we did on cadavers, some manipulations we were able to do on live animals while they were anesthetized, receiving veterinary care, as well as making sure that these met exceeded the range you would expect based on experimental data for the activities we were interested in. And the model has been parameterized in this way, so it should, with this range of motion, should be perfectly acceptable for studying bipedal and quadrupedal walking, as well as studying climbing. And the only caveat I will make is chimps have a highly mobile hip, and so it's possible you could have a particular climbing posture that might exceed the, say, hip flexion range of motion or one of the other points. And so usage of the model for studying climbing would need to be done cautiously. But from some of the climbing data we've collected, we think that many aspects of climbing can be studied with this model. You just want to be cautious for any trials where the animal put its hip, or knee for that matter, in a really extreme posture because it could exceed the validated range for the model. Final comment on this really large range of motion at the hip and some steps we took to try to address it is that when you take a joint through a substantial range of motion, what looks like a nice muscle pathway when the model is in the reference posture can become quite contorted and experience a number of different types of problems. You can have muscles, these red lines, that, for example, penetrate through bones or appear to penetrate through bones. If you're using these so-called via points, they can kink, so that if one via point that's supposed to be distal to another moves past the proximal one, you get kinking of the muscle path. Or if you're using wrapping functions, like we've done fairly extensively in the hip, the muscle can actually slip off of the wrapping function if you go through too large a range of motion. And any one of these, particularly the kinking and the wrapping, can introduce discontinuities into the muscle tendon length and moment arm functions, which are not going to work well when you do a simulation. And I've just demonstrated here using a commonly available human model this effect. So here in this model of the human musculoskeletal system, the hip is flexed, adducted, immediately rotated, and you can see the muscle is wrapping along an ellipsoid here to prevent it from passing through the bony structure of the hip joint itself. And from this top diagram to the bottom diagram, all I did was flex the hip one additional degree, and now the muscle has gone from the back part of that wrapping surface and slipped over the top, creating a discontinuity while also changing its moment arm. It now lies in front of the hip joint center instead of behind, so it would switch from functioning like an extensor to a flexor. And this is a pretty extreme posture for the human hip to be in. You're not going to see this if you're just simulating walking, but this was within the defined range of motion for the hip joint in this human model. And because we suspected that in future uses of the chimp model, you're going to have a much greater probability in getting into one of these extreme postures, we went to great lengths to try to prevent any of these issues. And so what we did was we developed an iterative procedure that took the chimp model through every possible combination of hip joint angles, flexion, extension, medial lateral rotation, and abduction in 1/10 of a degree increments, and generated these surfaces looking for discontinuities, and which, of course, we found in some cases. And it turned out in most instances, it was fairly easy to remove those discontinuities by making a very small change to the location of a via point or the shape of a wrapping surface. But we hope that this effort will pay off in future uses when you go beyond, say, using the model for just walking to, as I had mentioned earlier, future uses in climbing where there may be more extreme joint postures. There are a number of muscle model parameters that are needed to create a musculoskeletal model. And again, here there's a situation where there's not nearly as much data for chimpanzees than there is for humans, but there actually is data for chimps, whereas for many other species, these data just don't exist. And so muscle model parameters like optimal fiber length or peak isometric force were based on cadaveric dissections done by Thorpe and colleagues and Nagano et al. Some of the techniques that are used in human musculoskeletal models to determine the tendon slack length, so the resting length of the tendon in the muscle model for each of the muscles. A number of these techniques that are used based on average sarcomere length or matching peak isometric torques just currently are not possible in chimpanzees. So we used a numerical optimization procedure to estimate the tendon slack length based on a technique that was described by Manal and Buchanan. In terms of evaluating and validating the model, there's a number of steps we took. Some of the things, again, you would traditionally do in comparing a human musculoskeletal model to experimental data simply aren't possible in chimpanzees. We can't, for example, put the chimpanzee on a dynamometer and ask them to produce maximum knee extension torque at 10 different joint angles. It would be great if that was possible, but it's just not, unfortunately. But there are still some key outputs that can be subjected to a validation analysis, and one of those are the muscle length and moment arm functions. And so we compared the moment arms predicted by our musculoskeletal model with measured values done on cadaveric specimens from a study by Thorpe et al., and additional data that was collected by my colleague Matt O'Neill as part of this study, primarily to fill in some of the gaps for muscles that Thorpe and colleagues had not measured. And so here's an example of what this type of comparison looks like, and there's many more graphs you can find in the 2013 JEB paper. But this is the across knee flexion range of motion. This is the moment arm of the quadriceps and 2 of the hamstrings muscles. And the solid line is the model prediction, and the dashed line in each case is the measured value, the experimental value. And you see here that the model is capturing many of the key features of the experimental data in terms of the relative magnitudes and being relatively constant on average versus increasing with joint angle. And the main difference is that you've got some higher order characteristics, some curvature in some of the model predictions where there's just a straight line for the experimental data. And when you think about it, you would expect some of these functions to actually be curved, and they're going to depend on the cosine of the joint angle as that's changing. And so I would never be one to suggest that a model shows experimental data to be incorrect, but I think it's reasonable to expect that there probably was some curvature that simply was not picked up based on the techniques that were used in the Thorpe study. And at the end of the day, what we're mainly interested in here is that we get the approximate magnitude correct and also some of the trends in the data. Another aspect of moment arms that we were very interested in and did a fairly thorough analysis on that you can find in the paper was the effects when you have a spherical joint like the hip of changing one joint angle on the moment arm about another joint axis. And so what this graph shows you is the flexion and extension moment arm of a particular muscle, again, the gluteus medius, as you flex and extend the hip joint. Each of the individual lines in the graph represents a particular degree of medial or lateral rotation of the hip. And so you can see at some points in the hip flexion range of motion, the moment arm can actually switch from relatively large extension to relatively large flexion just based on medially or laterally rotating the hip. And so that is extremely important for interpreting the functional role of a muscle. And it's something that you just can't get a full appreciation for if you're ascribing the function of a muscle based on an observation of how an animal moves and maybe some EMG data to give you the timing of activation. You really need to have the overall 3D anatomy represented to be able to tease out effects like this and to identify which muscles you have a potentially large effect like this for, whereas other muscles were very insensitive to changing the off-axis joint angle in terms of the moment arm about the primary axis. We also did a number of sensitivity analyses on the model outputs. These were focused primarily on things like muscle attachment points and via points and wrapping functions that affected the pathway of the muscle, and then also the muscle model parameters. There's an extensive set of analyses in the paper. For anyone who's interested, I'll just give you one example here. It's well known that musculoskeletal models, the force output or the moment output is most sensitive to the tendon slack length. So of all the muscle model parameters, typically the highest sensitivity is to the tendon slack length. And this figure shows the sensitivity of the force produced by each muscle in the model, or a sampling of muscles in the model, to variations in the tendon slack length. And the blue bars are the chimp model and the white bars are the human model. And the main thing to note here is that for most muscles, when there's a difference, the blue bar, the chimp model, has a lower sensitivity than the human model. In some cases, it's pretty substantial, like here. In other cases, it's modest, as there. And so what this means is that the chimp model is actually less sensitive to tendon slack length than the human model overall. And this actually makes some degree of sense because it's been known for quite some time that the sensitivity to tendon slack length in terms of the force a muscle can produce is related to the relative length of the fibers versus the tendon. And one of the characteristics of chimpanzees is they tend to have relatively long muscle fascicles and relatively short tendons, whereas humans as a whole tend to have relatively longer tendons and shorter fibers. So this reduced sensitivity to tendon slack length is a good thing in terms of model performance because there's some uncertainty about the tendon slack length, but it's also a characteristic that makes sense when you think about the differences in muscle architecture between humans and chimpanzees. So just to put that workflow up here again and sort of summarize how we've gone from a skeletal specimen through the scanning process, model construction, and model evaluation. And as I've mentioned, for anyone who's interested in more details, they can find them in our 2013 paper. And if you want to, instead of just reading about it, actually try out the model, whether you might use it for your own research or simply curious to look at it and see how it is different, perhaps from human musculoskeletal anatomy, it is freely available on the SimTK website. If you go to Google and simply type in SimTK chimp, it's the first few hits that come up. So a couple of final comments related to the model and its development is about the time we were starting on this project, there was a new updated human musculoskeletal model that was published by Edith Arnold, Scott Delp, and colleagues. And we made a point as we were developing the chimp model where in cases where it made sense, we tried to make the chimp model as similar to the human model as possible in terms of the level of complexity and modeling decisions that were made, while still respecting the situations where there clearly was a difference based on the anatomy. And we made these steps in the hopes that it would facilitate comparisons that are made based on the human model versus the chimp model. And you can imagine that if you compare the outputs of two different models from two different species that are very different in complexity, you're not going to know how much of that difference is due to differences between the species or just differences in the complexity of the model. So we've taken some steps to try and make these two models analogous to each other to facilitate these sorts of comparisons. Also, I mentioned early on that a lot of the initial steps in putting the model together were done in the commercial software Sim, and this remains an excellent choice for doing this sort of work. Over the years, they've built up a number of features and capabilities that are really useful for building models from scratch. And the reason we went that route was because at the time, OpenSim, while it has great tools built into it for working with existing musculoskeletal models and running analyses and simulations, did not have a robust set of tools for building a model entirely from scratch. It certainly would be possible, but it was not initially designed with those capabilities. But looking to the future, someone else trying to reproduce what we've done with a different species or create a complementary chimp musculoskeletal model may be able to do this in open source software. So part of the roadmap for OpenSim calls for continuing to develop and expand the model building and model editing capabilities. And then there's also other complementary software, such as the open source NMS Builder, which stands for Neuromusculoskeletal Model Builder. And this allows you to build models based on imaging data, like you would get from CT scan, MRI, et cetera. And after completing your model, you can export it in OpenSim format and load it directly into the OpenSim software to use it. So I think as time continues to go forward, there will be very robust open source alternatives for building musculoskeletal models from scratch, as we've done with the Chimp model. And so just to wrap up, I'd like to reinforce this idea that musculoskeletal modeling is a really useful approach that's complementary to the traditional approaches that are used to study functional morphology and how it relates to behaviors like locomotion. And the ability to actually have the morphology represented in the model makes it a really powerful technique. In this vein, we've developed a model of the chimpanzee musculoskeletal system, pelvis and hind limb, and corresponding anatomy that's freely available to anyone who wants to use it. And our continued work with it is going to be done in concert with human modeling and simulation studies, and as I mentioned earlier, complementary simulation studies based on models of extinct human ancestors to really get a better idea of how morphology influences our movement patterns and locomotion and how this may be related to the evolution of human bipedalism. So I will stop there and turn it back over to Jim.
Yeah, thanks, Brian, for a great talk. Super interesting results and super clear as well. So now we'll have time to start the Q&A session. As I mentioned at the beginning, all the questions will be text-based, so you want to find the Q&A interface in WebEx. Go ahead and type in your question and make sure you select to ask all panelists. So let's see, while we're waiting for questions to come in, I thought that was really interesting that the Chimp model was less sensitive to tendon slack length. Are there any other parameters for which it was more sensitive, or was it just in general the same or less sensitive than a human model to some muscle?
It was tended to be about the same in the other parameters. And so some of them are not surprising, like the sensitivity. We were looking at the sensitivity of muscle force output to peak isometric force. You have a sort of a unique one-to-one relationship there. But for most of the other parameters, it was pretty similar. It was just that one that was lower in some of the muscles, certainly not all.
Mm-hmm. Okay. And then it's great that you've shared the model. I was wondering what kind of other materials come along with the model that people might be able to use to get started.
Right. So currently, and as you can imagine, this is part of a long-term project. Currently, we have the musculoskeletal model, so it includes the OSIM, the OpenSim file you need for the model, as well as all the bone files that give you the graphical representation of the skeletal anatomy. And we will be making, we've done a three-dimensional kinematic analysis using the inverse kinematics tool in OpenSim, comparing chimpanzees and humans. And so we'll be making those data available on the same website. And then as we continue to progress through our projects, we will make certain aspects of them available on the same Chimp model page. And so what will probably be among the most interesting and exciting is we're moving towards trying to do predictive simulations in chimpanzees versus humans. And as we do these, we'll continue to make more and more of the materials, either models that are scaled to individual chimpanzees as opposed to just a generic model, as well as the simulations that go along with them.
That's great. Thanks, Brian. So now we have some questions from the audience. So let's see, from Anahita Koshkai. Did you compare the variability of muscle parameters for a chimp versus human?
That's a good question, and it relates to the statement I made earlier, in that for a lot of species, these data simply don't exist. And for chimpanzees, we're fortunate that they do, but they are available in such small numbers that we don't really have a good sense of the variability the way we do for human muscle parameters. You now have human muscle architecture studies based on 20 specimens or more, and the two chimpanzee studies I cited were both based on one individual each. So it's something that's really lacking right now, and it really puts us where human musculoskeletal models were in, say, 1990, where we're dealing with very small numbers. And it would be great if in the future we have much larger data sets so we can have an idea of what the variability is and how it may compare with humans.
Okay, great. Now a question from Nidhi Sitapathy. For the extinct models, we have the bone structures from fossils, but how do you go about determining what the muscle structures are for extinct animals?
Sure. So for those in the audience who may not know, or this may not be immediately obvious, when you have fossil remains, you're usually left with a representation of a skeleton, but the soft tissues are almost never preserved. So you rarely have any of the muscles or tendons. And so the reconstruction of the muscles and what their relationship is to the underlying skeleton is based on, where possible, identifying attachment sites. Sometimes you can see the markings on the bones, although it's not always possible. And then making informed guesses or informed comparisons between extant species. And so most mammals, certainly all primates, have essentially the same bones in their hind limb, the same joints. They more or less have the same muscles, and these muscles tend to attach in reasonably similar areas. And so you can create a provisional model for an extinct species based on your general knowledge of primate musculoskeletal anatomy. And then what you would typically do is, in cases where you're left with the greatest degrees of uncertainty, let's say you have a case where there is a clear difference between, say, the human case or the chimpanzee case, you could do two versions of the model that look more like one case or more like the other. And then you would also really need to do some sensitivity analyses to see what aspects of your muscle attachment locations are the model predictions most sensitive to. And you would always take those results with a much greater grain of salt than you would when you're working with an extant species, where you can have a greater degree of confidence. And it's not directly in the context of a model of an extinct species, but a number of the sensitivity analyses we did on attachment points with the chimp model would be very informative in terms of where you would expect to have the greatest degree of uncertainty when you're reconstructing the muscle attachment points in a case where you really only have the skeleton, preservation of the skeleton to work with.
Okay, thanks, Brian. We should actually we'll have to go ahead and wrap up since it's almost 11 o'clock. If there are other questions that we didn't get to, we can forward those to you, Brian. So we can answer after the webinar. You could help me flip through the last couple slides. We'll go ahead and wrap up. So thanks everyone for the great questions, and to Brian for the really interesting and really clear talk about how to build a model and how to use a new model. Next slide. So I want to acknowledge the funding sources for OpenSim and the NCSRR. So we're supported by several grants from the NIH, including the NIH grant that funds the National Center for Simulation and Rehab Research. Next slide. You can find out more about the NCSRR, OpenSim, and upcoming events and resources on our website. And we also hope that you'll complete the survey that will appear in the pop-up window at the conclusion of the webinar, which will help us improve the webinar series and choose upcoming topics. I think there's one more slide, maybe. Nope. Oh. So with that, I'll just say thank you, everyone, for participating. Thanks, Brian. Thanks, everyone who tuned in and asked questions. We hope you'll continue to stay involved with OpenSim going forward. Thanks, everybody.